So in the parasite group, first we were studying about protozoans, right? In protozoans, we were done with the Sarcodina group and the flagellates, right? So what is left? We have the sporozoa left and the ciliates left, okay? So let's start. So first we are starting with the ciliates, okay? There's only one ciliate. What is the ciliate? It is Balantidium coli, okay? How do you identify Balantidium coli? You'll see a protozoan with cilia at its periphery, okay? So that will help you to identify. Look at the cilia. So this cilia will help you to identify Balantidium coli. Remember, it's the only protozoan which we are studying which has cilia. This is the largest intestinal protozoan. It will present with diarrhea and the host is the pig, okay? So those are the most important points about Balantidium coli. Now let's start with the sporozoans. Now these sporozoans, they can either be intestinal sporozoa, they can be tissue sporozoa, or they can be blood sporozoa, okay? So let us start with the intestinal sporozoa. Now intestinal sporozoa are the coccidian parasites, okay? Now these coccidian parasites are acid fast. What are the other acid fast organisms you know? There is Mycobacterium tuberculosis, okay? Then you have Mycobacterium leprae. Then you have Nocardia, okay? These are also acid fast. And then we have our coccidian parasites. Now this coccidian parasites, examples are Cryptosporidium parvum. It has been asked in your exam. Cyclospora and Isospora. So for these organisms, the image that they will give you is something like this, okay? So what are you seeing? I've always talked about acid fast organisms. They are pink organisms in a blue background, okay? So whenever you see pink organisms in a blue background, you should think about acid fast organisms. So these are acid fast organisms. This is actually the image for Cryptosporidium parvum, okay? Now the image for Cyclospora and Isospora is also pretty similar. The main difference between these three is the size, okay, is their size. So what do they cause? Now this is also going to cause diarrhea, okay? These are intestinal sporozoas. They will also cause diarrhea, specifically in HIV patients, okay? Generally when you talk about an HIV patient with pneumonia, what is the specific organism? Pneumocystis jirovecii, right? But what is the most common cause of pneumonia in HIV patients? The most common is still Streptococcus pneumonia, okay? And what is the specific cause of meningitis in HIV patients? It is Cryptococcus neoformans, and if you remember, that has the India ink appearance. Sorry, that has India ink is used for staining the capsules of Cryptococcus neoformans, right? And if there's a HIV patient with diarrhea, think about the coccidian parasites. Most important example will be Cryptosporidium parvum. It has been asked. Next you go to the tissue sporozoa. Now this tissue sporozoan, you have your toxoplasma. Now toxoplasma can have three forms, okay? Oocysts, tachyzoids and bradyzoids. Now all these three forms are infective, okay? Where do you get the oocysts from? The oocysts, you will get it from the contaminated food and water. Tachyzoids are these structures here, the sickle shaped structures, okay? These are tachyzoids. Now these tachyzoids are present in the blood, okay? They are present in blood. So it can be transmitted by blood transfusion, okay? Bradyzoids on the other hand are present in the tissues. So this is how it looks like. This is a bradyzoid. This is present in the tissues, and when you eat the meat, so basically you're eating tissues. If you eat infected meat, you can get the bradyzoids, okay? So all the three forms of toxoplasma are infective forms. Now the hosts, very important. The definitive host is cat, intermediate host rat, and accidental host man. You can remember if you watched, I think everyone's watched Tom and Jerry, right? So you have the cat and the rat, and sometimes you have the man, right? You can see a human sometimes in Tom and Jerry, okay? So cat, rat and man for toxoplasma. Now going to the blood sporozoa. Now in the blood sporozoa we have two. First we have the plasmodium, which causes malaria, and then we have babesiosis, okay? So first let us look at the life cycle. Now first we are looking at the life cycle of Plasmodium falciparum and Plasmodium malaria, okay? So when the mosquito takes a blood meal, it is injecting the sporozoids, okay? So the infective stage for man is what? It is the sporozoids. Okay, so sporozoites are injected. Now these sporozoites go to the liver cells. Okay, liver cells. In the liver cells, they'll infect the liver cells, they'll form the schizont. Okay, now whenever the schizont ruptures, you have the symptoms of malaria like fever and chills. Now this can also infect the, now this cycle which happened in the liver is called the exoerythrocytic cycle. Okay, now this can also infect the RBCs. Now that cycle will be called erythrocytic cycle. So what is happening? It is infecting the RBC. You have the immature trophozoite stage, the ring shaped form. Then you'll have the mature trophozoite stage. You can have the schizont stage, and then again the schizont will rupture and the cycle will continue. So sometimes what happens, the trophozoite develops into gametocyte. Okay, so all of these stages are seen in the RBC, erythrocytic cycle. Questions come from the erythrocytic cycle. When they show you a blood smear, like this, they are basically talking about the erythrocytic cycle, right? Because you can see the RBCs there. So what are the stages you're going to appreciate in erythrocytic cycle? You'll appreciate the trophozoites, the schizont, and the gametocytes. Okay. Now this gametocytes, obviously there will be male and female gametes. Whenever the mosquito bites again, it will take in the gametocytes. Okay. So the male and the female gametes will fuse inside the mosquito. That is called the sporogonic cycle. Okay. Male and female gametes will fuse inside the mosquito. And what will they form? They'll form an oocyst, and that oocyst will release sporozoites. Okay. So now which one is the definitive host? What is definitive host? It is the host in which the sexual cycle takes place. Okay. Now sexual cycle is obviously taking place in the mosquito, because in the mosquito the male and female are fusing. Okay. So the mosquito is the definitive host of malaria. Again, so you have the erythrocytic cycle, you have exoerythrocytic cycle in the liver, and you have sporogonic cycle in the mosquito. Where is the sporozoite? Sporozoite is inside the mosquito, okay, in the salivary glands of the mosquito. Then you have a trophozoite stage, schizont stage, gametocyte stage in the RBCs. Okay. Now, if you compare the life cycle of falciparum and malariae with ovale and vivax, one difference you will see is this, hypnozoites. Now hypnozoites are present only in the cycle of ovale and vivax. Hypnozoites, what are they? They are basically dormant forms. Okay, they are dormant forms of the parasite which are responsible for causing relapse. So it's a very common question that relapse is seen in what? Relapse is seen in infection with ovale and vivax. Okay. Now some questions. What is the infective form for the man? That is sporozoites. Who's the definitive host? The female Anopheles mosquito. Intermediate host, humans. And transfusion induced malaria, that means by the blood, is caused because of the trophozoite. Okay. Now let us compare the 4 forms of malaria: falciparum, ovale, vivax, and malaria. Now there are some dots, okay, which you need to memorize. Morer's dots are for falciparum, ovale has James dots, vivax has Schaffner's dots, and malaria has Z-man dots. You can remember Manila Zoo, okay, so malaria, Z-man. You can remember orange juice, okay, so ovale, James. And falciparum is Morer's dots, okay. Falciparum is Morer's dots. You can remember that falciparum ends with M, okay. So you remember Morer's dots. Okay. Now, falciparum affects all ages of the RBCs. All ages of RBCs. So first you should know that young RBCs are larger in size. Okay. Example, I'll show you. This thing you see in this diagram, you see this RBC is larger than the surrounding RBCs. So this RBC is a young RBC and there's infection in young RBC. Okay. Compare this with this. So here you see an RBC which is infected, and this RBC is smaller than the surrounding RBCs. So this is an old RBC. Okay. So young RBCs are larger in size, old RBCs are smaller in size. falciparum affects all ages of RBCs. Now the key thing you need to remember for falciparum is it causes multiple infections. What does that mean? See, look here. Inside the RBCs, we have 3 trophozoites here. We have 2 trophozoites here. So whenever you see multiple infections, we are talking about falciparum. This is the schizont stage of falciparum, and this is the gametocyte stage of falciparum. So what is very easily identifiable is the gametocyte, which is banana shaped, and the trophozoite, okay, which has multiple infections. Okay, so whenever you see multiple infections, you can straight away answer falciparum. Whenever you see the banana shaped gametocyte, you can answer falciparum. Now, how to identify ovale? Now, to identify ovale, first of all, it infects young RBCs, okay, and the RBC is fimbriated. So this one word is enough to identify ovale. Okay, remember fimbriated RBCs. So you look at this RBC, you can see these fimbriae here. You can see the fimbriae in the schizont stage. Okay, you can see these fimbriae here. Okay, you can see fimbriae here. So whenever you see fimbriae, you can identify ovale. Okay. How to identify vivax? Vivax also infects young, but it does not have fimbriae. Okay, so I can see a large RBC, so it is young, right? If there's a large RBC, that means it's a young RBC. So I know this RBC is young. I know this RBC is young, but there is no fimbriae, so that means it is vivax. Okay, so RBCs are young in ovale and vivax. Okay, RBCs are young in ovale and vivax. So whenever you look at an RBC which is larger, you should think about ovale or vivax. Okay, ovale will always be fimbriated, so vivax will be a large RBC but not fimbriated. Okay, now that's how you identify vivax. Okay, how do you identify malaria? First of all, malaria will infect old RBCs. Okay, old RBCs, so they'll be smaller. So look at the RBC, it is smaller than the surrounding RBC. So this is most likely malaria infection. And these band forms, okay, they are specific for malaria, Plasmodium malariae. Okay, band forms. And in the schizont stage, the pigment deposition will be in the center, and then you will have these the appearance of a rosette. Okay, so if you have a rosette appearance in the schizont, that is definitely malaria. Okay, so what is specific for malaria? First thing, the rosette appearance and the band forms. Okay, so if you see band forms, small RBCs, okay, small RBCs, band forms, rosette, that is for malaria. For vivax, large RBC but not fimbriated. For ovale, fimbriated. And for falciparum, multiple infections and banana shaped gametocyte. Okay, that should do it. Now see, this is a blood smear. Okay, these are the blood smear. Now these blood smears, they can either be thick blood smears or thin blood smears. So a thick blood smear is taken to know the presence of parasite. It will just tell me if the parasite is there or not there. Okay, it will not tell me which parasite. Okay, thin smears are used to know the species. Okay, thin smear is used to know the species. Now you can learn this. Okay, what are the conditions preventing falciparum infections and conditions preventing vivax infection? Let's start with the next one, Babesia microti. This is also a blood sporozoa. Okay, blood sporozoa. It causes babesiosis. Okay, transmitted by tick. It has a Maltese cross appearance. So look at this appearance here, very important. Okay, for babesiosis, remember Maltese cross appearance. Okay, transmitted by tick. Now this is about the protozoans. Okay, this was about the protozoans. So let us go to the table and see. So in ciliates, we have only Balantidium coli. Okay, in sporozoa, you have blood sporozoa, which includes Plasmodium, falciparum, malaria, vivax, ovale, and you have Babesia microti. Okay, for tissue sporozoa, you have Toxoplasma gondii. Now Toxoplasma is what is the presentation? It can be asymptomatic, but if it is symptomatic, it can have hydrocephalus as a presentation, especially in children, and it can cause posterior uveitis. Okay, it is one of the examples of the diseases which cause posterior uveitis. Intestinal sporozoas are the acid-fast organisms, coccidians, right? Cyclospora, Isospora, Cryptosporidium, Parv. So this is about the protozoans.